Thank you guys for coming. Thanks for inviting me. You guys, I'd like to announce that the next AbSciCon is going to be at Georgia Tech. And it's going to save us a lot of money in airfares and hotels. These are all the Georgia Tech people at this meeting, or a lot of them. There are a lot of them here. Okay, I'm going to talk about biopolymers, and you guys, my time doesn't start yet because this is kind of background information. This new mission, you know, the Dragonfly, this is like so good for us, right? We're going to a planet with polymers and organic chemistry. This is going to be really exciting for all of us here. Okay, so I want to talk about polymers. You know, like, does biology actually need polymers? And are biopolymers special among polymers? And what can we learn about biopolymers, you know, about the origin of life? What do biopolymers actually tell us about the origin of life? And when we think about biopolymers, of course, this is sort of where we start, which is really, you know, this is information flow in biology, right? There's sequences of DNA. That's information is transcribed to RNA. It's translated to protein, and then protein actually does things with that information. So this is all about information, and information is sequences of monomers that are linked. That's really essentially what polymers are about. So isn't it really important to me, feature of polymers is that they are all made by dehydration condensation. Okay, every universal biopolymer is made by essentially the same chemistry, which is you take two monomers, you put them together, and a water molecule comes out. And there's a lot of other things that are universal. They all go through phosphorylated intermediates. They're all made by processive motors that use divalent cations. There's a whole lot of universalities in biopolymer synthesis, but I want to focus on this water element here. This is the way I think about it, is the way I think you should think about it, which is that you know, a metabolite is a small molecule that is either used as a substrate or a product in an enzymatic reaction. And water is clearly the most abundant and frequent and universal metabolite in all of biology. You use over your body mass of water in chemical reactions every day. So water is the primary metabolite of biology. What does that mean? If we think about the surface of the land, now I'm talking about Nick Hud kind of ideas here, that's sort of where they come from, is the activity of water on the entire planet cycles every day. So reactions that take up and release water are pushed and pulled on the entire planet every day, except in certain places, and that's in large bodies of water. That is a place where the activity of water is constant, and we're not going to be able to push and pull reactions by this process. So the abiotic synthesis of polymers by the cycling of water is a metabolic process. And think about what Moran was saying if you were here for her talk early. You take monomers, you dry them down, you make polymers, and you bring them back up into solution, and they're kinetically trapped. So what you've done is you've taken sunlight, effectively, and you have incorporated that energy into chemical bonds. Okay, so that is basically what the dry down reaction does. It's taking environmental energy and it's putting it into chemical bonds. And you repeat this process, and you can keep adding more and more energy into chemical bonds. Okay, so this is metabolism to me. And it's kind of funny. I remember in the old days, Steve, and people would have these arguments about what was first, nucleic acids or metabolism. And the problem with those discussions, I think, was that the way people were thinking about metabolism was in terms of the Krebs cycle running in reverse and things like that. But if you think about metabolism in this way, I think it changes the argument. I think Steve might, I haven't talked to him about this, but I think he might accept that metabolism could take a lead before biopolymers if we define metabolism like this. You don't have to say anything. I'll ask you later over beers. Okay, so now let's talk about, actually it's kind of ironic that the people who finally discovered this metabolic process were nucleic acid people. They were Nick Hud and Ram Krishnamurti and people like that. So the people who are focused on metabolism are really interested in the Krebs cycle and redox chemistry and things like that. And they really weren't paying attention to simple chemistry, which is hydration, dehydration, which is near equilibrium processes on the surface of the Earth. So if we look there, then we can see that it's really easy to imagine metabolism and biopolymers, coming together. Okay, so this is a polymer. Polymers are amazing. They're magic. Okay, polymers are magic. That's the thing you have to realize. They're undescribably amazing. So if you look inside those things, what you can see is this beautiful self-complementarity. All biopolymers are self-complementary. If you look in cellulose, all the donors donate and all the acceptors accept. If you look in the center of a protein, the donors and acceptors are beautifully aligned. And of course, if you look in nucleic acid chemists are used to talking about complementarity, but that complementarity extends to all biopolymers. Okay, every biopolymer is self-complementary. You can see anti-parallel beta sheets and parallel beta sheets and alpha helices. Everything is beautiful. Everything is perfect. Okay. The problem is the same thing is true in ice. If we take a water molecule and we crystallize it, and you look in that crystal, you'll see the same thing. Small molecules, when they form crystals, are also self-complementary. All the donors donate and all the acceptors accept. So the question is really, what is special about a polymer? How is an ice cube different from a polymer? What is the fundamental difference between these things? Okay, this is the difference. If let's say I take a mixture of 20 amino acids and I very slowly cool it, they will resolve. Okay, this is called recrystallization. People make money off of this, right? This is how you purify things. You recrystallize them because monomers will resolve into simple systems. Biology doesn't want that, right? Biology wants enzymes, informational molecules. It wants to, through evolutionary process, put functional groups at specific places in three-dimensional space. Okay, that is not allowed here. Everything is determined by this small molecule. So there's no control of that biology requires. So we want to do this, but nature doesn't let us do that. Okay, if I take 20 amino acids and I try to assemble them, I can't do it. If I take guanine and cytosine together in water, they don't form base pairs unless they're polymerized. Okay, the magic is in the polymerization. So this doesn't work, but this works. Okay, this linking things together, and the linkage is really important. Okay, it does two things. First, there's the proximity. It keeps things close together, so the entropy is much less forming this thing because these things are already constrained to be close together in space. So there's a big entropy argument. But the other thing is, in biopolymers, these are not flexible strings, right? These are very specific. For example, a peptide, highly restrained, pre-organized, and really already pushing very hard toward this assembled thing. In fact, any RNA, you make any sequence of RNA except for a homopolymer, and it will assemble. Right? RNA just wants to assemble so bad you can't stop it. And any sequence of polypeptide will form beta structures. Right? So the assembly of these molecules is built into the backbone in many ways, and it's just totally inescapable. People complain that they make random sequences of polypeptide and it always comes out of solution. The reason is that it forms beautiful beta structures. Right? Those aren't just random precipitates. Right? Those are assemblies that have been designed by nature over long evolution. How much time do I got?
We got 6 minutes.
Oh, okay, good. So we talk about proteins spontaneously folding. Right? You take a protein in solution and it will spontaneously fold. But the only reason for that is that you're cheating on the accounting. If you really add everything up, like how much did it cost to make the amino acids? How much did you invest in selecting for the right sequence? And then, of course, for making the bonds in aqueous media, proteins do not spontaneously fold, right? They only spontaneously fold is because you're starting with the polymer, okay? So, and that's kind of cheating thermodynamically. So this is, I think, the way to think about it. This is what nature can do really easily. And what nature has done is it has separated the cost of the assembly. Nature wants complex assemblies. It wants informational biopolymers. It wants catalytic sites. And it has separated the cost of that assembly from the making of the assembly. So the sequence of this thing and the backbone has been designed by billions of years of evolution, right? That is an investment that nature has made in this polymer. It has selected the side chains, it has selected the sequence, and then what actually does the polymerization, of course, it uses ATP and hydrolyze. So there's an enormous amount of energy that is put, and the reason that energy is invested is in order to make assemblies. And so this is the analogy I use. Money. If you don't have to pay for stuff, you can have everything, right? And that's what nature has done. It has invested so much in the past that assemblies are free. And so the way to think about it is if you have saved your whole life for your kid to go to college, then you can send your kid to college essentially free because you have that account. And you don't have to pay for it in real time. That's what biology has done. Biology has been investing in assemblies for billions of years and has made them spontaneous and free. Right? This is why nature does such crazy things, because it really doesn't have to pay for it. Right? It's already paid. That investment is a done deal, and everything it wants to do is essentially free. So, thank you guys. This is my group. Actually, this is Loren, who has done a lot of work on the peptide stuff. This is Anton, who talks to me about these things all the time. And the other member of my group are here. Thank you, guys. And
Okay, we can only do one quick one because I've been informed that we have another group coming in right after us, okay? So, yeah, you usually disagree with Loren, so I hope you're going to.
He does.
He didn't say anything about human beings.
Oh.
I thought it was an interesting idea about the water activity being like the first metabolism. When I talk to RNA world people, they usually don't talk about metabolism in that world. But you need some type of energy to replicate yourself. So is water activity variation a candidate for the metabolism in an RNA world?
I would say no, because RNA is so far from equilibrium that these processes, in fact, I would say RNA is a very derived polymer that comes from a lot of chemical evolution. If you look at peptides and sugars and other things you polymerize, you can do them near equilibrium by dehydration. It's very simple. RNA, because it's so far from equilibrium with that phosphate, that's the only polymer that it doesn't work. So I would argue, based on that and some other things, that RNA is a very derived polymer, and of the polymers we have, it's the one that is furthest away from the sort of origins of chemical evolution.
All right, great, thank you. Let's thank you.